Hi, so this podcast is on Mendel's laws and also the dihybrid cross. So the results from the monohybrid crosses that Mendel did showed that there were inheritable factors or genes that are responsible for characteristics. Now remember that Mendel didn't know about DNA, so he called them factors. Mendel determined that the phenotype of an organism is based on the genotype of the organism, and that each trait is based on two genes, one coming from mom and one coming from dad, and that true breeding individuals are homozygous. They have both alleles, which are the same. So that brought up the law of dominance. The law of dominance is one of Mendel's first laws, and it says that in the cross of parents that are pure breeding for contrasting traits, only one trait's going to appear in the next generation. So in other words, all offspring will be heterozygous, and they're going to express the dominant trait. And the law of dominance can be shown here if we're looking at yellow and green P colors. Yellow being dominant over green, and we see that in the F1 generation, or when we cross our F1s, we get a 3-to-1 ratio. The law of segregation. The law of segregation is Mendel's second law, and the law of segregation says that during the formation of gametes, which are eggs or sperm, the alleles responsible for traits separate from each other. They separate because of meiosis, if you think back to meiosis. They're then recombined at fertilization, producing the genotype for the traits of an offspring. So if we think of meiosis and we think of gametes separating and then coming together during sexual reproduction, bringing about a new genotype for an offspring. The law of segregation can be seen here in this diagram. If we have an organism, this would be our female parent, and our female parent producing eggs. The eggs can either be this genotype or this genotype. Similarly with the male parent, the gametes there could either be this genotype or this genotype. And then these right here in our Punnett square, these are the possible combinations for offspring. And we would see, if we had a lot of offspring, we would see a phenotypic ratio of about 3-to-1 for this particular genotype here. And so this demonstrates the law of segregation. The law of independent assortment. The law of independent assortment says that alleles for different traits are distributed to sex cells independently. Mendel didn't just stop with one trait. He wanted to know, if I trace two traits at the same time, how do those two traits interact with each other? And this was done using a dihybrid cross. And so this is the next thing that we're going to look at. The dihybrid cross was a breeding experiment that tracked the inheritance of two traits. It shows Mendel's law of independent assortment. It shows that each pair of alleles segregates independently during gamete formation. And the formula to sort of calculate the alleles is this right here. Now this should be 2 to the power of n, not 2n, where n is equal to the number of heterozygotes that we're looking at. So for example, we've got some practice here just to sort of apply this formula. Remember, for some reason this didn't show up properly, so that should be 2 to the n. So in our first question, big R, little y, there are 2 heterozygotes. So if we go 2 to the 2, that equals 4 possible gametes. And so this is a first step when determining how many gametes I need to account for in a cross. So a couple more. We have 3 heterozygotes here. This is a homozygous dominant right here, so we wouldn't count him. So that would be 2 to the 3. Eight gametes. And number 3, I want you to try. Pause the podcast and give number 3 a shot. See me if you need some help in figuring it out. Okay, so in a dihybrid cross, if we're looking at seed shape and seed color, Mendel took his classic 9 and he started a variety of crosses with them together. So if this was a cross that we're looking at right here, um, the first thing that we would sort of need to do is from there we would need to figure out what the gametes would be. So remember, we can apply our formula to figure out how many I would need. I have 2 heterozygous, so I need 2 to the power of 2 or 4 possible gametes. So now I go about creating combinations. These combinations would represent what would happen during meiosis. So for this particular offspring here, one possible gamete would have a big R and a big Y. One possible gamete has a big R, little y. One possible gamete has a little r, big Y, and one possible gamete has a little r, little y. So these are all the possible combinations. Now the other parent, that'll be the same. So big R, big Y, big R, little y, little r, big Y, little r, little y. So that's the first step. The first step is getting the gametes. Once we have the gametes, we put them into our Punnett square. And now our Punnett square is going to look a little bit different because we are looking at a dihybrid cross. Okay, so we've calculated out that we have 4 possible gametes. Again, I put the parents on the sides. Now, if we look at this dihybrid cross here and we look at this Punnett square, we no longer have 4 squares to work with. We've got a much larger one. A couple rules. I'll do the first line and then I'd like you to pause the podcast and try to fill in the rest. Start with one letter first. Dominants always go first over recessives in a letter. And be consistent. Start with R's in this case and continue with R's. Okay, pause the podcast and continue to fill that in. And again, if you have any questions, come see me. So what you would have found after doing that is the following genotypes here, and there's a diagram showing their phenotypes. What Mendel noticed, being the mathematician that he was that phenotypically we find a 9:3:1 ratio for these phenotypes right here. Okay, and this was a very classic phenotypic ratio that demonstrates independent assortment, that the alleles are, for 2 traits, are moving independently of each other. And that brings us to the test crosses. So of course we can do test crosses. Here we have an individual with an unknown dominant trait, and we want to determine what that trait is. Whether it's heterozygous or homozygous dominant. We can do that in a test cross, and remember the test cross is taking the homozygous recessive. So here's an easy way to do one of these Punnett squares. If you come across a Punnett square here, we know that one of our traits, whatever it is, we know that this one here is got just one possible gamete. Right? Because the only combination there is little b, little c. So really what I'm looking at as far as a Punnett square, this one too, so little b, big C, and then there would be 2 gametes here, possibly little b, and then something else. This is a very easy Punnett square to figure out. So the next slide takes us through the 2 possibilities. So if this was a big C, we would find that we would only have one possible gamete. And so after filling it in, we only have one genotype. If this was a little c, well, now we have two possible phenotypes for our plant. But again, making the Punnett squares easier allows you to do that, and that way you don't have to do those huge 16 square Punnett squares. So to summarize Mendel's three laws, the first law was the law of dominance. The law of dominance was determined by our parental cross, and what we found was that when we cross two contrasting traits, we get offspring that are showing what's known as the dominant trait. The law of segregation, remember that alleles segregate. That can be seen in our monohybrid cross. And the classic ratio for there was our 3-to-1 ratio, 75%, 25%. Independent assortment was that if we use two traits, and we were looking at two different types of alleles, that those alleles will segregate independently, and of course we get this 9:3:1 ratio. This is the end of the podcast. At this point, you can go on and try the dihybrid crosses.